Welcome back to the deep dive. Today, we're plunging headfirst into a classic yet utterly devastating problem in the world of biopharma scale-up.
It's the moment a process that looks like a total superstar in the lab
Right.
absolutely collapses into an economic black hole at the pilot scale.
And our sources today give us a really specific, almost painful case study. It's all about microbial fermentation, focusing on a production strain of Corynebacterium species.
This failure is just a perfect illustration of what happens when you treat that bench-scale success as the finish line.
Exactly.
So the goal today for you listening is to really understand why a process achieving truly excellent upstream titers, and we're talking phenomenal, like 18 to 22 grams per liter.
Which is a huge win.
A huge win. But it was rendered completely unfeasible. The process looked great right until the overall recovery yields just cratered.
From what to what?
From an anticipated 75% all the way down to below 40%.
Oof. And that's the core mission for us today, right? To figure out why the system was so broken. Because here's the kicker.
The cells were doing their job.
The cells were doing their job. They were making exactly what they were supposed to. This wasn't a biological failure. This was a physical, economic disaster. And it was driven entirely by scale.
It's the definition of a disconnect. It's a failure of what we call upstream, downstream integration.
Yep.
And we have to uncover why that high titer alone is not enough to define success, and why a seemingly benign factor broth rheology, how thick and gooey the liquid is, why that emerged as the pivotal critical quality attribute, the CQA that sank the whole project.
I think we need a mental model here just to frame this whole conversation. We're not just talking about a technical glitch.
No, not at all. Let's
Yeah.
establish it right up front. High titer is the starting gun. It is not the finish line.
Okay, I like that.
If you ignore the physical properties of the broth you're creating, you are imposing what we're calling a hidden tax on the whole process.
The hidden tax.
Yeah.
Imposed by scale.
Exactly. Scale doesn't just make the vessel bigger, it reveals this tax by forcing these nonlinear changes in fluid dynamics that just amplify the worst parts of the cell's physiology.
Okay, let's jump right in then. Section 1, the bottleneck. Let's get tangible. What exactly went wrong when the process jumped from, say, 5 or 10 liters in the lab up to 300 or 500 liters?
Well, the shift wasn't gradual. That's the key here. It was catastrophic.
Yeah.
Really abrupt.
Abrupt.
Yeah, it happened specifically around the 72-hour mark in the large-scale fermentation. That's when the apparent viscosity of the broth just surged.
And we're talking a little bit thicker.
No, we're having a minimum 3- to 4-fold increase compared to what they saw in the lab. The minute the total suspended solids got past about 18% weight per volume, the broth fundamentally changed.
And when a fluid fundamentally changes like that, it invalidates everything the engineers thought they knew, doesn't it?
Correct. The broth transitioned into what we call a non-Newtonian fluid.
Which, unlike water, means its viscosity isn't constant.
Exactly. Specifically, it started showing this severe shear thinning behavior. It's also known as pseudoplasticity.
So it thins out when you mix it fast, but then it gets thick and gel-like when it's just sitting there moving slowly through a pipe.
You've got it. And that characteristic completely invalidated all the equipment sizing, which was, of course, based on the assumption of a predictable Newtonian flow.
Wait, I have a question. If the broth got so thick, why couldn't the downstream engineers just, overpower it, run the motors faster, pump harder?
And that's the classic engineering challenge. And the hidden tax is the answer. When you try to force a fluid like that through equipment designed for something simple like water,
You break it.
you don't just increase the flow, you destroy efficiency, you increase wear, and the specific downstream failures were just brutal.
So tell us, what happened to the core separation steps, the really critical stuff?
Okay, so first, centrifugation. The efficiency just plummeted by over 50%.
50%.
At least. The disc stack centrifuges, which are supposed to separate the cells, were immediately and severely fouling.
From what? All that gunk.
From cell debris and, crucially, from massive amounts of these extracellular polymers.
And fouling means you have to stop.
It means frequent, unscheduled shutdowns for manual cleaning. You just can't run a continuous process if you're stopping every few hours to degunk the bowl. That's labor, time, and just lost yield.
And I'm guessing filtration, which is next, must have been awful.
A nightmare. An absolute nightmare. Depth filtration throughput completely collapsed. They projected 150 liters per square meter.
And they got
Under 40. The high solids load and this sticky gel-like matrix just clog the filter media instantly.
Wow, so the filters become basically a wall.
An instantaneous barrier. The cost and time and disposables just skyrocketed. They were changing out filter trains multiple times per batch.
And I imagine even the final really expensive step, chromatography, felt the effects of this.
Absolutely. The highly viscous feed streams cause these excessive differential pressure spikes in the very first cycle.
And that's a bad sign.
It's a clear indication of physical stress on the column. The feed was literally squishing the expensive resin bed, causing compaction. The net result of all these compounding failures.
The yield dropped below 40%.
And the batch turnaround time got extended by 2.5 days. Economically, the process was dead on arrival, despite those amazing upstream numbers.
Okay, let's move to section 2, root cause analysis. We know how it failed physically, but why? Why did the cell culture suddenly turn into sludge at 500 liters when it was perfectly fine at 5?
It was a disastrous synergy. It was biology reacting badly to engineering stress.
Okay.
And the main biological culprits were these stress-induced adaptations, which were triggered by late-stage nutrient limitation.
Specifically nitrogen.
Specifically nitrogen, at the pilot scale.
Yeah.
In those larger, less uniformly mixed vessels, it's just fundamentally harder to keep nutrient gradients consistent.
So the bacteria are stressed out because their food supply is getting patchy. And what does Corynebacterium do when it feels that kind of stress?
It immediately switches into defense mode. The cells ramped up their exopolysaccharide secretion, EPS.
These are the sticky polymers.
Complex, sticky polymers. Polysaccharides like glucans or mannans, they form this protective, high-viscosity matrix around the cells. Biologically, this is linked to known stress signaling pathways. The bug is basically activating genes to build its own protective, gooey fortress.
That is genuinely fascinating. So the cells are just trying to survive what they think is a harsh environment.
Right.
And in doing so, they completely sabotage the entire industrial process. It's like biological self-sabotage. But that wasn't the only thing making it thick, was it?
No, correct. Compounding the problem was secondary metabolite buildup, which, combined with all the other stress, triggered partial autolysis.
Cell self-digestion.
Yeah, the cells started to break down, releasing all their intracellular components into the broth, proteins, lipids, and, critically, DNA.
And why is DNA such a problem? We just think of it as genetic material.
Released DNA is a powerful viscosifier. It's highly charged. It forms these long, entangled viscoelastic gels that just drastically amplify the thickness and the fouling.
Especially when it's mixed with the EPS.
Exactly. So you have the EPS building the structure and the DNA cementing it all into this impenetrable matrix.
So now let's look at the engineering factors that created that stress in the first place.
Yeah.
The larger vessel must have altered the fluid dynamics dramatically.
It did. At the pilot scale, the engineering solution was really a compromise to keep the dissolved oxygen, the DO, above 20%.
Which is critical for the cells.
Critical. But without causing too much shear from the impeller, they had to use really high gas sparging rates.
So lots of bubbles.
Lots of bubbles. This compensated for their power constraints, but it introduced heterogeneous microaerobic zones.
Pockets of the reactor where the oxygen level dipped too low.
Exactly.
Okay.
And those pockets stressed the cells, which fostered subpopulation variability, and that just worsened the stress response and triggered the whole EPS cascade.
This just highlights why scale-up is so incredibly tricky.
Yeah.
You fix one problem, like shear.
And you create another one, like nutrient gradients and stress zones.
Right.
And this feeds directly back into the rheology modeling. The Broth's pseudoplastic rheology is modeled by these complex power law equations. The key thing to know is that their models assumed a simple Newtonian flow, which was just fundamentally wrong.
So the sources are basically screaming that for predictive scale-up, you need specialized work. You need to do things like computational fluid dynamics, CFD modeling.
They have to
really match your power input and your oxygen transfer rates. If you don't do that modeling, you're not scaling up, you're just making the problem a lot bigger.
You are flying completely blind. And we should briefly mention the other factors, too. They were using fixed-time harvesting.
So just pulling the batch at a set number of hours?
Right, which completely overlooked these dynamic changes. They didn't have the right process analytical technology, no torque sensors to measure viscosity, no capacitance probes to see when lysis was starting.
And even the raw materials they used played a role.
They did. They switched to a more cost-effective soy hydrolysate.
To save money.
To save money.
Yeah.
But it introduced inconsistent high levels of polysaccharides and, critically, metal ions like calcium and magnesium.
And what did those do?
These ions actively cross-link the EPS chains, which significantly enhances the gel strength. It was the biological equivalent of pouring superglue into your broth.
Wow. Even a simple switch to a cheaper raw material had this devastating cascade effect. Just incredible. That brings us to section 3, the real case and the organizational fix. This was a mid-sized specialty chemicals firm.
Right, they were scaling a C-glutamicum strain for a cosmetic active secondary metabolite.
Let's talk about the human element of this failure.
The human element is so critical here. The lab batches were spotless. But after 3 failed 500-liter batches, management was on the verge of just abandoning the strain entirely.
Even though the product itself was perfect.
Exactly. And this mirrors challenges across the whole industry, really. But the key failure here was organizational. The teams were heavily siloed.
Meaning the upstream fermentation team and the downstream purification team, they just weren't talking to each other effectively.
Not at all. The upstream development team was judged, and their bonuses were tied purely to hitting that 18 to 22 GL titer. They were hailed as heroes.
Right.
Meanwhile, the downstream processing team was seen as a separate cost center, almost an afterthought, and they were forced to deal with this unprocessable nightmare. They were being judged on the recovery yield.
Which was tanking because of what the upstream team did.
So the upstream team gets the bonus for high titers, while the downstream team takes the blame for the economic failure of the entire process.
That is the classic organizational pitfall this source material is highlighting, isn't it?
It is. Biological stressors at scale will always amplify engineering flaws, and that pressure point is always at the interface between teams. The solution couldn't be just technical, it had to be about systems thinking and integrated PT.
You have to measure the properties that matter to the next step, not just the ones that make your department look good.
That's it in a nutshell.
Okay, let's move to section 4, the integrated technical solutions. So the core realization was you couldn't just use brute force downstream. You had to go back and target the root causes upstream to stop the catastrophe from happening at all.
That's right. The first big step was a comprehensive media reformulation, specifically targeting that nitrogen limitation stress.
And how'd they do that?
They moved from a single, fast-releasing ammonium sulfate source to dual nitrogen sourcing, which incorporated urea.
What did that change do physiologically for the cells?
Well, urea modulated the release kinetics. It sustained ammonia availability over a much longer period.
So it's like giving the cells a slow-release nutrient patch instead of just one big meal and then starvation?
That's a great analogy. And that sustained availability averted the stress signal that was triggering the EPS secretion. Less stress, less goo.
A beautiful biological fix. What was the second chemical fix?
They tackled the raw material problem by adding trace metal chelators, specifically EDTA, to the media.
And how did that, untangle the goo?
The EDTA acts like a molecular bodyguard. It selectively binds to those free calcium and magnesium ions, the ones from the cheaper soy, and it sequesters them before they can interact with the EPS.
So the metal ions can't form those crosslinks.
Exactly. So the gel structure collapses, which actively reduces the overall viscosity and the gel strength.
That's a crucial two-pronged attack from the very beginning. What about how they controlled the fermentation and when they decided to harvest?
They moved away from that arbitrary fixed-time harvest to a condition-based harvest.
Using new tools.
Using new real-time tools. Viscosity indices, which they measured by the torque on the impeller, and capacitance probes to actually detect the onset of autolysis. They harvested only when that trade-off between maximizing titer and avoiding the rheological collapse was perfect.
A smart, dynamic approach.
And they also made a small tweak to the seed train, an extra expansion step, which helped minimize inoculation shock.
So finally, what if a little bit of EPS was just unavoidable? What did they do right at that interface between upstream and downstream?
They implemented a small, very controlled post-harvest enzymatic treatment.
An enzyme?
Yeah, an enzyme like polysaccharidase. It goes in and it just snips the bonds in the EPS polysaccharides. That alone dropped the viscosity by about 35% without degrading the final product.
And that was the tipping point.
That was the tipping point that made centrifugation and filtration viable again.
That is just such a powerful combination of biology, chemistry, and engineering. It really shows why integrated design is so necessary.
Absolutely. And this success brings us right back to Section 5, the mental model and the economic validation. So for any R&D scientists listening to this, here is the mental model you need to carry forward. High titer is not the finish line. It is the starting gun.
Right.
That hidden tax of viscosity gets imposed the moment you scale up. That comfortable 5-liter process you're running. It's an idealized bubble. Scale unveils hidden physiologies like these stress EPS links, and you have to anticipate that.
So for the R&D scientist working at, 1 to 10 liters, what's the critical advice? What should they be doing now besides just tracking titer?
They need to integrate rheology metrics early. Don't wait for the pilot plant to scream at you.
So tracking simple things.
Simple things. Standardized pipette flow times, maybe some basic viscometry to detect early trends, and they must use design of experiments DOE to actively probe those stress points.
Stress the cells on purpose to see what happens.
Exactly.
Yeah.
Mimic the scale effects you know are coming. Define your harvest windows based on recoverability, not just the theoretical peak titer.
And the outcomes validated this whole approach. The economics were massive.
They were. Downstream recovery yield jumped from that failure rate of below 40% all the way back up to 72%.
Which is huge.
And because of the reduced downtime, the fewer consumables, the cost per kilogram of product dropped by a staggering 28%, and batch variability tightened to less than 10%.
Those metrics are just an absolute validation of unified design. You fix a biological problem in the fermenter to solve an engineering failure in the centrifuge.
And we connect this to the bigger picture, embracing this unified design and comprehensive P8, along with that crucial cross-functional collaboration. It can potentially avert 20 to 30% of bioprocess failures across the entire industry.
This case wasn't unique.
Not at all. It was symptomatic of how deeply siloed metrics can sabotage success.
So the lesson is clear. Rheology has to be treated as a CQA from day one. Scale doesn't just make things bigger, it reveals the complex, stress-induced responses of your organism, and it forces you to think about the entire system.
And this raises, I think, an important question for you, the listener, to take away. In your own field, what hidden physiological or physical metric are you currently optimizing for? A metric that looks perfect at the bench scale.
But which could secretly be sabotaging the next critical step of the process.
Exactly.
That's the kind of systems thinking that saves companies millions. Thank you for joining us for this deep dive into biopharma's hidden scaling challenge. We appreciate you trusting us with this complex material.
Thanks, everyone.
We'll catch you next time.